Hey again, this is ZengoMD. Well, the human's best friend has always been our dogs, and why not? They reduce our stress, are just so cute and furry and fun. But not everyone has a pet or has a lifestyle where they can keep one. But I'm going to nominate another living thing or group of living things that all of us own as our pets. So let's find out why the living inhabitants of our GI tract are probably our real best friends and find out what we can do if they're sick and what kind of treats we can give them to keep them happy. Let's look first at where these cute little living things reside. Well, the stomach's up here and it's mainly filled with some really strong acid. Well, they don't like living there very much, so we won't talk about that much anymore. Therefore, we're going to focus mainly on the small intestine here, where most nutrient absorption occurs, and the large intestine, where most of our water balance in the GI tract happens. The absorptive surface of the small intestine close-up reveals many folds studded with villi that add to the vast surface area, and along each villus we see tightly connected cells that make up our absorptive surface called the gut endothelial lining. Above it all lies a layer of mucus and bacteria that is anything but random and anything but passive in its relationship with the GI tract cells, our immune system, and even our brain. But first, the GI tract has been one of the most woefully misunderstood systems over the last 50 years. Here's an example. Acid, an important part of the stomach's functioning, was actually implicated in ulcer disease, creating a multi-billion-dollar acid blocker industry until we discovered that ulcers were caused by a bacteria, forever changing the way we treat this common ailment. Well, the GI tract gets blamed for all of our problems, like that pesky gas, bloat, or our nervous stomach. Here in the small intestine, we didn't worry about how to make the good bacteria happy. We worried about when it got reduced to the point, like taking antibiotics, where it got reduced enough so that bad bacteria could overgrow. Yet we really never understood the good bacteria we were supposed to have. The large intestine was a source of Crohn's and inflammatory bowel disease, and a place we need to scope for polyps and cancer. Otherwise, we took it all for granted. We really didn't understand the function. We just assumed it was this passive absorptive surface. Mainly because all we had to really see the diversity of organisms was a culture, meaning we had to take stool or another sample and wait for things to grow. And this failed to identify most of that intricate variety. We felt that the brain controlled the gut by making it go slower or faster, but the gut had no say back to the brain. And oh, how the GI tract was our enemy when we had a colicky baby, too. In the last 10 years, we've been able to look at ribosomal RNA found in all bacteria and identify thousands of different strains of our single-celled friend present in healthy people. We've learned how these little cells communicate, how they protect us, and even how they induce changes in our brains. The absorptive surface can be as large as a tennis court and have 2 to 10 times more cells than all of the functioning somatic cells in our body. You know, the cells that come from our mother and father. Yet this world inside of our gut weighs only 7 ounces. The mucus biofilm layer that lines our intestines has a structure to it. The outermost layer that interfaces with our food and drink directly contains a majority of our friendly microbes embedded in a mucus layer. Then below that is an additional mucus layer that protects our sensitive absorptive cells from toxins, irritants, and allergens. Below that is a layer of immunoglobulins that can bind up harmful antigens before the actual absorptive surface of our GI cells is finally reached. If these cells are well protected and healthy, they serve as the final selected barrier. And that's shown when their junctions between the cells are tight. If they are not, these tight junctions, the last line of defense, begin to loosen. This leads to increased permeability for stuff we don't want to absorb that will find its way into our bloodstream. This is referred to as leaky gut. But as you can see, in order for the gut to get leaky, there need to be multiple layers of failure in our gut biofilm, mucus, and local immune response. Like when our friendly house guests are invaded by pathogenic bacteria that don't belong, and this punches holes in that primary line of defense. It really does kind of look like a dotted line. But before we get into leaky gut and what can go wrong, let's first take a look at the top 10 coolest features of the gut biofilm. And I guarantee you, once you're done looking at this list, you will want to do everything you can to get rid of the bad bacteria and give your good bacteria all the treats that it needs so that it's happy. Here we go. Number 10. It forms automatically starting at birth. We have a sterile GI tract when we're born, or we have minimal exposure to bacteria. But these guys just move in and start paying us rent. Vaginal deliveries are exposed to more bacteria. Therefore, the gut biofilm forms faster than C-section babies and breastfed babies tend to form faster than bottle-fed, but generally by year 1 to 2.5, we have an adult distribution. However, most skin rashes in infants are associated with a gut dysbiosis at an early age. So you get this skin indicator that things in the gut are not going well at fairly early on. So don't ignore it. Next, they make the vitamins you need, like B12, folate, pantothenic acid, right where we can absorb them into our system. Now, they're certainly not the primary source of these important vitamins, that's diet, but it is certainly nice of them to do that for us. Number 8. In fact, even their waste products are useful. The excess products of the gut biofilm form here serve as food for the gut biofilm and the colonic cells down here. These are known as short-chain fatty acids, and they are one of the most hotly researched topics in GI medicine right now. And we can even test for them in your stool. Number 7, it supports your immune system. There are many relationships between gut bacteria and the secretory IgA immunoglobulin produced by gut cells. One is to help the gut bacteria form a physical barrier with the IgA molecule to protect the endothelial lining. These short-chain fatty acids also can regulate gut production of these immunoglobulins and even secrete toxins to kill unwanted gut bacteria before they are even presented to the endothelial gut skin surface. Pretty cool. Next, they participate in signaling to each other and signaling to the gut endothelial cells. This communication is truly remarkable in that they work in tandem when needed to achieve a benefit for you, their kind host. We just mentioned how the two-way signaling involves the production of the immunoglobulin SIgA that is expressed in the lumen of the intestines. You know, the gut has its own little immune system. They can also mutate under stress and even engage in gene transfer between each other in order to adapt to the changing environment in the gut. That's pretty incredible. You know, your big dog can't just mutate into a small dog because you moved from the country to an apartment in the city. Next, changing gut diversity correlates with increased aging, inflammation, and disease. So is the secret to managing aging in your gut biofilm? People who maintain a more youthful gut microbiome tend to have fewer diseases as they age. Here's a link between gut biome and Alzheimer's and Parkinson's. There are also links to autism and chronic pain, in addition to the well-known link with GI disorders and GI symptoms. Next, staying with the brain, there's also evidence that the gut biofilm is associated with depression, even altering expression and production of the most important neurotransmitter, serotonin. At least as far as depression goes, it's most important. And even though 90% of the serotonin is produced in the gut, it can still influence the expression of serotonin made in the brain. Serotonin is not the only neurotransmitter expressed by gut bacteria in the gut lumen, but it is also influenced by gut stress in the brain. GABA, the main anti-anxiety neurotransmitter, is associated as well. And finally, gut bacteria influence folate production and absorption, where folate is a major source or raw material needed for the production of neurotransmitters. Folate also becomes an issue if a patient has one or both of the MTHFR genetic mutations. Therefore, people who have that mutation but don't know it can really help their symptoms by ensuring that the gut biofilm is diverse and well populated, especially with the very common bifidobacter that helps to regulate folate. Next, it can normalize the stress response. Well, we've known for decades about how stress negatively affects the GI tract, but most of that has been done on direct nervous system and how it influences gut motility. We know that microbial diversity can also change dramatically when we're under stress. In fact, even in young children, exposure to early stress can change the gut biofilm for the worse permanently. However, when the gut is normalized through treatment, either fecal transplantation, now we're not sticking stool inside people yet, but we do it in animals, or any other means that support a healthy biofilm, we find out that the stress response is more likely to normalize quicker than if the biofilm remained dysbiotic. And you may remember from our video, A Fresh Look at Stress, how hard it is to turn off that chronic stress response, which can drag us down and make us foggy and fatigued. Number 2, it can protect against type 2 diabetes and even obesity by changing our feeding behavior. A healthy gut biofilm primarily prevents too many calories from being absorbed in the first place by presenting less food to the gut cells. But specifically, these short-chain fatty acids like propionate, made when your gut biofilm is well balanced and there are few intervening pathogens, work to make you feel fuller faster and resist overeating. Also, it is well known that obese individuals have less than ideal gut microbiomes compared to normal weight controls. So anything you can do to restore your gut biome should make any weight loss attempt more successful. Diet pills like phentermine and protein shakes do nothing to help the gut biofilm and will make any attempt at permanent weight loss even more difficult. Number one, you have complete control over how well these little guys behave. They listen better than your kids or your dog, and they won't take a dump in your house ever. Here we see how a lack of dietary fiber changes the microbiome to let more destructive pathogens have a friendly environment to live and prosper and punch holes in your biofilm. But if you make an effort at eating better foods, you can change your gut biofilm pretty quickly. The emergence of genetics in the gut biofilm and absorption is important, with the testing of genes like FUT2 and others involved in the histamine response in the gut. But here it is noted that even if you are genetically challenged in this area, you can overcome that with an improved diet. Look, this is the hottest idea of study in bioidentical medicine right now, and our approach to a patient today may be very different than 6 months from now, so stay tuned. For now, here are 2 great review articles if you want to dive deeper into this subject matter. The second review article is the source of this summary diagram showing the interrelationships between the gut microbiome and the brain directly, but more importantly, the gut microbiome's ability to protect the fragile GI endothelial cells from everything we eat, which directly reduces the stress that's transmitted up to the brain. A dysbiotic gut can result in more continuous stress impulses to the brain than a burn on your skin or a chronically painful joint, yet you may never know that daily stressor is constantly there. So what can you do? Of course, the best thing is to get the most up-to-date testing and a thorough genetic analysis. If that's not possible, some people turn to 2 kinds of supplements, probiotics and prebiotics. Probiotics are well known, however, many of them often have big television advertising budgets, but they're very low potency and low diversity. A fresh probiotic with species diversity and a colony count of at least 10 billion is recommended to change the population here. But how do you know which strains you are missing without a test? Also, the positive effects will reverse when the probiotic is stopped without any long-term dietary changes. See, if you're eating a bad diet, all the probiotics you swallow will have a minimal transient effect, and then they'll die off and end up in the stool. Prebiotics consist of soluble fiber called MAC, which stands for microbiota accessible carbohydrates. Instead of giving you more cells, it works to nourish the ones you want to keep. However, again, if your diet is horrible, then you're giving a lot more nourishment to the bad guys than a little spoonful of fiber is going to help your good guys. Speaking of diet, check out our video on conquering food addictions to learn how to eat to support your gut biofilm. It's actually the same thing. And speaking of food addictions, one reason why that diet works is because it increases the direct connections of the gut-brain axis to induce a feeling of fullness and reduce the neurotransmitters involved with food addiction. But more importantly, this natural food menu every meal without exception is the best way to strengthen the gut biofilm, so it serves as a better barrier to pathogens and toxins here at the gut lining. And absorption of fewer toxins is a good thing, but so is the reduced stress input to the brain and the endocrine system as a result. In all of my years of recommending this menu of whole, unprocessed protein sources, fruit, vegetable, and eggs, I have yet to find a patient who follows it and does not have more energy, less pain, less gas, and sleep better after really just a couple of weeks. So there you have it, how to take care of your few billion little house guests who work so hard for their meals and their safe environment by protecting you. Well, we need to learn a lot more. We need more information on what an optimal gut biofilm is and if that really should change with age. We need more studies on distant effects of the gut biome in humans, not just mice. And we need to learn the optimal way to treat imbalances of bacteria and imbalances in short-chain fatty acid production that will work and will continue to stick. We need to learn how helpful probiotics are long-term, and if there is a benefit to taking a designer probiotic formulated to your own deficiencies. So stay tuned to this channel and we'll bring you more on Human's Best Friend and how you can train it and give it the right treats.